Thank you for listening to this presentation about the soil health benefits of cover crops and forage systems. My name is Veronica Acosta-Martinez. I'm a soil microbiologist and biochemist at the USDA-RS in Lubbock, Texas, and I will be presenting results from the collaborations that we've had with Shock West, from the Department of Plant and Soil Science, Texas Tech University in Lubbock, Texas, Ghimire, Assistant Professor in New Mexico State University in Clovis, New Mexico. Soil health is the capacity of the soil to function within its ecosystem boundaries and the physical, chemical, and biological component. With soil health initiatives, we're just also trying to focus on the biological component, listen to the biological component, and see the soil as a living system that provides so many essential functions to all of us. Establishing indexes of soil health requires to understand the complex interactions of management, inherent soil properties, crops, and environment. And the biological component is one of the most sensitive indicators of changes in management and in these different factors, especially the top three, the bacteria, fungi, and protozoa. The microbial component are linked to essential soil functions as they are involved in 80 to 90% of the decomposition processes. So anytime we change crops, we incorporate cover crops, or add organic amendments and do other changes in management, it will impact the microbial component and their enzymes. And if they are improved, there will be increases in soluble nutrients in soils and in soil organic matter in terms of quantity and quality, specifically the composition, the chemistry. Changes in soil organic matter could also further improve the microbial component and provide better conditions to host a more diverse microbial community and bring, for example, increases in the fungal component, increases in the arbuscular mycorrhiza fungi that will host microbial associations with plants that can increase plant, water, and nutrient uptake, and that can help and give more resilience to that soil to more frequent droughts and climate variability. With changes in soil organic matter, there will be also improvements in aggregate stability and soil structure, and potentially increases in water holding capacity of our soils. So there will be improvement in many functions, including soil water storage and erosion control. The southern high plains semi-arid region experiences high wind speeds and hot temperatures, more consecutive days in a year, together with a lower average precipitation, less than 20 inches generally, and even less and less. So it is a challenging region to sustain soil health, but it's not impossible. It's just that this region, shown here in this square, is one of the most persistent dust-producing regions of North America. So these are the type of dust storms that we can see that resemble the 1930s conditions. So our soils are inherently low in soil organic matter because of the extreme climate in this region, and more frequent now and extremes, record droughts and heat waves that these soils are exposed to. With management under monocultures, there will be more winter fallow periods that can leave the soils more exposed and prone to wind erosion. So with the transition to dryland that we're experiencing in the Ogallala Aquifer region, we could change from the possibility with irrigated production to have high moisture and biomass production, and potentially increasing more the soil organic carbon of our soils, to the opposite on the dryland conditions with lower moisture and with more fallow periods, then lower biomass production and the potential to increase soil organic carbon. So the soil health will become more and more important to improve the capacity of these soils to hold more water from precipitation. So our management selections are important. If we could combine the use of organic amendments, cover crops and crop rotations, conservation tillage practices as possible, and livestock integration, we could improve more the soil health. Another option in this region, or just a program that is important for this region, is the Conservation Reserve Program. Texas leads the nation in this program. And what we need to consider is that all these benefits that are reached with this program, then in order to conserve them when we are changing and going back into crop production, it's going to be then also more important if we can do these more conservative management practices for our soils. Things that we cannot change are the soil types, the climate variability that we're experiencing, and the Ogallala aquifer water level. So one of the projects that we have is looking at this transition to dryland production in Texas. So we were able to identify 3 producers in Littlefield that were assisted by NRCS in changing from center pivot irrigation some of their land to subsurface drip irrigation. And we also then identify some of their land under dryland conditions, and we're able to take soil samples within the first year and second year of this transition. We also found a CRP site within the same soil type, and found, just even visually, you can see how the soil is looking more aggregated compared to the cropland. And it also has 10 times higher arbuscular mycorrhiza fungi populations and 3 times higher microbial community size than the cropland. So it is an ideal site that we can use as a reference point of the potential maximum increases in the microbial component that we can reach, and how any changes in management that these producers are able to do, how it could compare to the CRP. We're reaching here a lot of land and 13 different sites or scenarios to do comparisons in these sandy soils under cotton production. So the microbial community composition have shown already to differ in the transition to dryland after 2 years only. In this type of plot, you can see how the community composition under center pivot is different, is separated from the community under subsurface drip irrigation. There is also another scenario in which we were comparing another subsurface drip with dryland, and there was also then a different community on that subsurface drip versus the communities under dryland. So what is happening here is that we found decreases in the microbial community size in the microbial biomass carbon with this transition from center pivot, look at the numbers, to the subsurface, to the other subsurface, and to dryland. There are 2 times higher microbial biomass carbon in the center pivot compared to the dryland. However, we also found that in that dryland, the producer used a winter rye cover crop, and having that management history is showing 2 times higher the levels of the microbial biomass carbon than the nearby subsurface drip system. So what this is telling us is that it is possible that if more conservative management practices are selected as possible, including that management history with a winter cover crop, there could be a possibility here to compensate, to improve the microbial component versus those decreases that we're going to see from the center pivot to the dryland transition. We have another study with incorporation of sorghum and cover crops in dryland cropping systems, sandy soils, and this was within 5 years. The interesting scenario here was that during those 5 years there were 2 droughts in 2003 and 2006. And just visually you can look at the pictures of the forage sorghum and see how there has been for these crops, grain sorghum also and cotton, variations in the biomass that has been produced due to these differences in the climate. For example, in 2003, precipitation was just 9 inches in total. And you can even visually see there even the cotton, maybe more visual. And then the next, the biomass produced was so low in all these different type of crops. The next year in 2004, precipitation received was 27 inches. And you can then also even visually and with the numbers there of pounds per acre see how much biomass was produced. So also this affected the establishment of the winter cover crops every year. And our study involved a comparison of continuous cotton with cotton grain sorghum rotation, with cotton and rye grain sorghum rotation, and forage sorghum rye without cotton for 5 years. And what we found already after 3 years, the cotton rye grain sorghum rotation had higher microbial biomass carbon than the continuous cotton. But it took 5 years for the cotton grain sorghum rotation to differentiate the microbial community size from continuous cotton. So that rye was helpful in improving the microbial component, even under dryland. So in 5 years, if you look here at this graph, we had in the forage sorghum rye almost 3 times higher the microbial biomass carbon compared to continuous cotton. And then by then, both systems, the cotton grain sorghum rotation and cotton rye grain sorghum rotation, had 2 times higher microbial biomass carbon compared to continuous cotton. So showing that even under dryland and these extreme conditions of the climate variability, it was possible to improve the microbial biomass carbon in this sandy soil, sand-Alton soil with 68% of sand content. So we measure all these different soil biological indicators in year 5, and including the activities of many enzymes produced by microbes. And these trends were also found. The next year, all these systems were placed on cotton. And what we found is then a linkage between the activities of enzymes produced by microbes to soil productivity. If you see this positive correlation of the activity of beta-glucosidase involving cellulose degradation, carbon cycling, and phosphatase involving phosphorus cycling, with the cotton yields found the next year when all these systems went into cotton. So if you measure only the cotton yields as individually, there wasn't that significant differentiation among these cropping systems, but the relationship is showing that correlation. It's showing that link of the improvement in the activities of enzymes that provide nutrients in soils to the cotton yields of the next year. More recently, producers are transitioning to no-tillage in this region. And it is very important that we also look at these sensitive indicators, which are the biological component in this transition to no-till. We can also already see in 2 years on the no-till system under wheat that this producer is actually using as a cover crop, earthworms. And we have found other beneficial soil health changes within these 2 years of this transition. For example, if you look here at the microbial community size, microbial biomass carbon is 2 times higher in the CRP that we also identified in this other group of sites that we're working with. sandy soils too, and it's higher compared to the cropland. Especially here we can see the conventionally tilled side, but we can see how the no-till drip and no-till pivot systems are showing higher increases in the microbial biomass carbon compared to conventionally tilled site. And it's more obvious the distinction within these sites, especially the no-till dryland compared to conventionally tilled sites with the microbial biomass nitrogen. There is another study led by Rajan Ghimire from New Mexico State University in Clovis, New Mexico, in which he is evaluating different types of cover crops and mixtures, and I've helped with the soil microbial component. And in this case, what we're showing here is a figure showing the first bar for the soil under fallow, and how in only 3 years the use of other cover crops like pea, oat, canola, and in mixtures that you can see here with canola, oats and canola and pea, and another mixture that is more than diverse with other cover crops as well, is increasing the fungal component with different markers that I have been using when you extract fatty acids from soils. And specifically we also can see that higher increases in the fungal component were found under oat and under mixtures. So these early trends and changes in the fungal component are very valuable to be possibly detected, as you could maybe select better the type of cover crops that you want and redirect your management selections. So improvement in the fungal component, as I was discussing before, can bring so many improvements in the composition processes. With the AMF, improve plant, water, and nutrient uptake. Can bring also improvement in aggregate stability, and so many other improvements in soils. So the 4 soil health principles are shown here, as some of them have been addressed already with our previous studies. Minimizing disturbance with different ways of reducing tillage. Maximizing continuous living roots with crop rotations, cover crops, and forages. Maximizing soil cover with all of these systems and trying to do the residue retention as possible, and maximizing biodiversity is possible, bringing livestock and the rotational grazing involved. This is also another type of study that Rajan Ghimire leads in New Mexico State University in Clovis, in which he's also looking at the restoration of grasslands, the livestock integration in cropping systems to maximize the benefit of conservation practices with the forage production and use, and the winter grazing in standing crop or cover crop residue and grazing in native pasture for the rest of the year. So one of these studies and findings already using enzymes produced by microbes involving nutrient cycling are showing distinctions in native grass under graze and non-graze conditions, and it's also showing how it's so much greater the activities of this enzyme and others that also were evaluated compared to the cropland, more than 100% higher. It's also showing within the croplands that were evaluated that this wheat sorghum fallow rotation that had grazing is showing twice much higher the level of the activity of this enzyme involving carbon and nitrogen cycling compared to the other rotation evaluated of corn sorghum under no-till and under strict tillage. And it's due to possibly also differences in the crops, but also it's the showing the grazing benefits. If we evaluate soil organic carbon by itself to look at organic matter dynamics and have this type of information, that enzyme activities can also provide, we wouldn't see the differences in native grass due to grazing, or these different also improvements we found due to the wheat sorghum fallow rotation under grazing. So it is important to have sensitive indicators of soil health in order to do our management selections this early. Another of the studies that we are working on with involved in different forage species and mixtures in forage livestock systems to improve soil health and enhance water conservation and sustainability of this region. And in the first system that brought the livestock integration to this region evaluated old bulbous stem with Vivian Allen and showed benefits in the microbial component as well as the decreases that we found in water use and in nitrogen fertilizer. And then shockwaves have been evaluated for the past decade the introduction of alfalfa in this region into the old bulbous stem. And it also showed further reductions in water use is possible in dryland and the elimination of the nitrogen fertilizer. And then we also found in this research with the PhD student and now postdoc Krishna Bandari benefits in the soil microbial component. So in this figure, the one in the left is for microbial biomass carbon, and at the top 5 centimeter depth we found the highest level of microbial biomass carbon with this old bulbous stem alfalfa system compared to other forages like the old bulbous stem alone, alfalfa, and native mix. And it's significant then when we evaluated the microbial biomass nitrogen. I also wanted just to point with this red line the levels of the microbial biomass carbon that we have under continuous cotton after 10 years, and show then how many more times the microbial biomass carbon can be with forages. There is also this black line for the rotation that was used for this integrated first system that was evaluated here, that had wheat, it had rye, it had cotton, and a fallow period. And it shows then how much more the microbial biomass carbon can be improved with forages. There are at least 3 sensitive indicators for soil organic matter dynamics and stabilization that I wanted just to describe, as this region have soils with low soil organic matter, and also they're exposed to these hot conditions that do not help to accumulate soil organic matter. So we need to do more work on our soils and select more conservative management practices as possible. And it's good if we can look at more sensitive indicators of the changes in the carbon content of our soils, like palm is a partially decomposed type of materials that we can evaluate, and also we can evaluate POX carbon, which are more simple carbohydrates, amino acids, and sugars in our soils. And the microbial biomass carbon that I was describing, that is only 1 to 3% of soil organic carbon, but that it is more active, labile, and changes faster due to management. And these different type of carbon sources can give us earlier indications of potential improvements in soil organic carbon, in carbon sequestration. So they represent, as you see here, this triangle, the more active and labile carbon sources in soils that could be just within days and few years of being improved, compared to then if you use soil organic carbon. So in summary, establishing these linkages between the microbial component to the essential soil functions, especially water conservation, is critical for improving the sustainability of agriculture in the Southern High Plains. So these improvements on the microbial component, overall soil health, is possible with adoption of management practices that can diversify cropping systems when possible, the use of cover crops or crop rotations, or reducing soil disturbance, adding organic amendments, integrate livestock in the crop production. Any change, anything we do in 1 year matters when possible. We know all the limitations that are also needed to be considered, the economics, the climate variability, and it's just how, if it's possible to apply any of these conservative management practices, we could just try to be just improving our soil's health and be more prepared for future challenges of climate variability. We have in our team doing soil health assessments within the Ogallala Aquifer region a technical note available at the ogallalawater.org, and also our contacts will be available for any questions that you have, and producers that would like to work with us and have some more information about your land and changes in the soil health, to please contact us anytime, and we'll be happy to always work with you. And we want to thank then all the different teams from Chuck West, Rajan Ghimire, and my team also, and the producers that we're working with for allowing us to take soil samples, talk to you, and to find if we are able ways to help to improve your soils, and to all of you for listening to this presentation.